This podcast was produced by OAR FM Dunedin with support from New Zealand On Air.
The Science Notes podcast on OAR FM Dunedin, brought to you by the Division of Sciences at the University of Otago. I'm Dave McMorrin. Over the next half hour, I'll be chatting with a graduate science student about their research, their passion for science, and the things that fuel their creativity. Well, hello there and welcome again to Science Notes. My name's Dave McMorrin, and this time our guest is, I'm going to practice, Julien Eschenroeder.
Yeah, that's correct.
There you go. I tried. Welcome. Thanks for coming along.
Thanks for having me.
Julien is doing a PhD in geology at Otago University. Almost finished it.
Yeah, pretty much. In a couple of months I'm going to hand in.
Very good. So deep in the depths of the writing and the analysis and such. So geology, and in particular you're measuring amounts of stuff out in the environment with a view to trying to reconstruct and understand how things have been in the past. Yes,
that's right.
And how is it then that you find yourself doing this at Otago?
So I'm, as you may hear, I'm originally not from New Zealand, I'm from Germany, and there I studied geosciences and had more of a focus into environmental geochemistry. And at the end of my master's, I didn't want to write anymore one day, and I decided to have a look what to do after the master's. And then I found this project advertised online here in Otago. I never heard of Otago before, and so I applied for it, and came with a little bit of a delay because of the pandemic in New Zealand.
Yeah, it's been a theme of late, actually. A lot of guests who are handing in now are the ones who all started around about the pandemic. And
true.
and their first 6 months was actually just reading literature rather than doing anything and stuff. And
sorry.
But you got here. So whereabouts in Germany are you from originally?
I'm from the southwest. That's close to the border of France, in the Upper Rhine Valley.
Okay. And so you came all the way across the other side of the world.
Yeah.
And you found yourself here. So the project that you did here, it's similar to what you were doing previously, or?
No. So I changed my background, basically, or my specialization. I changed it with every step of my academic career ladder. So my bachelor, I focused more on geophysics, and then I changed into hydrochemistry.
Right.
And for my master's, I looked at sinkholes in Mexico.
Right.
And how microorganisms in these waters affect the water column and can lead to the formation of underwater speleothems, so underwater rocks.
Right.
And
so you got to go to Mexico for that?
Yeah, I did.
Oh, nice. Excellent.
Yeah, I was lucky. Just before the pandemic.
Right. But you didn't get stuck there?
No, but lockdown started 2 weeks after we came home.
Gosh.
I had a lot of time to write then.
Yeah. And so then you came out here, and so now you're doing something different again, right?
Yes. Now I'm looking at past climate and past environmental health in New Zealand's lakes, specifically in the Southern Alps of New Zealand. And I'm trying to figure out how past climate variability over the last, let's say, 4,000 to 10,000 years has affected these lakes.
Right. So the lakes that you're looking at are, it would have to be said, not conveniently accessible. I had not heard of them. I looked them up online and discovered that they are in absolutely the middle of nowhere. So for the listeners, whereabouts are your lakes?
They're as much in the center of Fiordland, of the Fiordland National Park, as you can basically get. So if you would see Fiordland National Park as a teardrop, and you draw...
So this is, assuming we've got people from overseas, this is the southwest corner of the South Island of New Zealand, where there are no roads.
There are no roads, there are only mountains. On the west side there are fjords, on the east side there are lakes, gigantic
and maybe
lakes.
moose?
And maybe moose, you don't know, it's an ongoing question. And so if you would draw two lines as a cross over this district, you find the lakes quite in the middle.
Yeah. So it's miles. For people who know Manapuri, It's sort of close to that.
Yeah, if you follow one of the arms of Manapuri and follow the valleys across, then at some point you would maybe come out in the mountain ranges where those lakes are. Yeah.
So how do you get there then to do your sampling and stuff?
Usually by helicopter.
Right.
That's also what we did. So I participated in one sampling campaign in April 23 to resample one of the lakes and the catchments. And for that, we actually had to fly in with helicopters. They transport all our gear, which has to be, of course, and also very light. And we spent, I think, 2-3 nights in a hut that's close to one of the more secluded tramping trails.
Right. So there are huts and stuff.
There are huts, yeah. That was quite lucky.
Yeah.
I didn't know that would be the case. But yeah.
So why then are we looking at lakes in the middle of nowhere?
So my project comes in like the periphery of a very large New Zealand-wide study, Lake 380, that ran out by now, but they basically looked at how lake health, they assessed the lake health status of New Zealand's lakes.
Right.
And one of the aspects was to see how does human interaction affect the lakes, how does eutrophication form, where do we have problems with water quality. And then the range where I am more working in is looking at how natural climate variability affects those lakes, especially now when looking at the ongoing and intensifying global warming, how that will affect lakes in New Zealand.
Right.
And to make sure that we don't have any human signal in our lakes, we go as far as possible away and look at the healthy natural conditions and see how those lakes formed and developed and played or reacted to the effects of climate forcing.
Yeah. So in some ways, it's almost like doing a control experiment.
Yeah.
For all the other ones. The other ones are looking at the human effect. You're trying to find out what all the things that were going on or were going on, regardless of whether people were there or not. So you can assess the difference that the human impacts made.
Yeah, you can describe it as such.
Okay. So then, so you went in, you flew in, you stayed there. Hopefully it wasn't too cold. It probably was.
It was a little bit cold, but we had very good weather, actually.
And so you were taking water samples or?
No, also we take water samples to understand better the modern system of the lakes. We take soil samples, plant samples as well, even some because of the deers that are now all over the lake, or this lake area. We take feces samples,
right?
But the main focus is really on surface sediment samples and the cores.
Right.
So we brought coring equipment and took cores up to 3 meters sediment depth.
Right. So this is big long tubes of dirt down into the earth. 3 meters long and what about 20 centimeters wide or something, are they?
Yeah, that's pretty good estimate.
I'm guessing from what I've seen, pictures and stuff. And so the point of that is to do what?
The point of that is to look at, so we take the cores, we bring them back to Otago.
Yep.
And here we have a core facility, OCA, the Otago Repository for Core Analysis.
Excellent.
And there we open our cores, and then the first thing we do is scan them with so-called XRF core scanners. XRF stands for X-ray fluorescence. Sorry. I also have problems pronouncing that. And with this scanning devices, we can see the elemental composition of the sediment in the tubes on up to 2 micrometer scales, and how that changes. So that gives us already a first idea on sediment provenance, on possibly changing climate variability, because let's say when we have more rain and surface runoff into the lakes, we get more, we get heavier, like simplifying, we get heavier elements into the lake, like, such as titanium. So titanium would be then an indicator for surface runoff in this case, and we could say like, it rained in this part of the core apparently more.
Yep.
And
So it's a bit like, I mean, like the sort of geology people might have seen, where you have rocks and there's sort of striations in the rocks, and you can relate that to different eras in the history of the rock and stuff.
Exactly. Yeah.
Same idea, but you're going down into the ground to do the same thing.
Yeah. Mm-hmm.
And so there must be some way then that you can say, okay, a meter down into my core, I have an extra amount of titanium.
Mm-hmm.
You must have some way of correlating that with how long ago that was.
Yeah. So for that, so the course that I took now last year, I took samples in certain like distances down the core, and then I visited ANSTO in Australia, the Nuclear Science and Technology organization there, and there we did a radiocarbon analysis on the sediments.
So carbon dating them.
Carbon dating, exactly.
Okay.
And with that, you get back, of course, your results for these specific samples, and then you load them into a model, and that can give you then an age-depth relationship.
Right.
And from that, I can see then should have been with a certain uncertainty in this time, which is great because that enables me also then to compare to other records all over the world, or specifically more interesting in New Zealand, to see if my signal as I observe it is actually or has been observed in other parts of the country as well.
Right, yep. Because other people have done similar sort of coring exercises in other parts of New Zealand.
That, or they have completely different proxies.
Right.
Such as pollen analysis is very common in New Zealand. And from that you can also get similar aspects of the hydroclimate as it changed.
Right. So you can measure how long ago these different layers were, and then you have ways to relate what you're seeing at these different layers to what's going on with the weather.
Yeah.
And that's the main focus, is it?
In one of the main focuses, yeah.
And so your 3 meter core then, how far back in terms of time does that go?
So the 3 meter core that we took in this specific lake, it reaches until the end of the last declassiation in this part of New Zealand.
Right.
And we dated it at around 17,000 years ago. That with uncertainties, of course, that is approximately how far we can go back in time in this area. Everything before would have been there would have been maybe a lake, but that would have been just sitting in front of a glacier, and not a lot of organisms would have lived in this lake, and so we can't really give that specific sediments then a date.
Yeah. So 17,000 years ago, most of Fiordland would have been ice.
Yeah, or the glaciers were just retreating, leaving the first lakes to develop.
Yep, and then you can start getting dirt and sediment and stuff forming, and then you start your core, right? Yep. So you said that, I mean, and I'm guessing that the University of Otago, I mean, I don't want to assume, but I'm guessing the University of Otago didn't pay for the helicopter. This work is done in association with all sorts of other organisations, like you said, the people in Australia and stuff as well. So it's a much bigger exercise.
So the people in Australia, that was a complete different cooperation, because I got a postgraduate research award to work with them.
Right. Okay.
And then the helicopters, they were the rest funding of the outgoing Lakes 380 project. So that was the nationwide project, and there was GNS involved with.
Geological nuclear sciences, which is the big organisation in New Zealand that do this sort of stuff.
Yeah, one of, I think, two.
Yeah. Oh, well.
Yeah, I have to mention the others as well.
Yeah, fair enough. So there's, and indeed you've sent me an abstract of a paper, and there's people from all over the place. There's people from Germany there as well.
Yeah.
So it's quite a big. It's not the sort of work that you can do just you and your supervisor in the lab by yourself.
No, you need quite specialized instruments.
Lots of toys and gear and equipment and stuff.
Yeah.
The Australian lab allowed you to work out the age by doing carbon-14 analysis, but there's other sorts of analysis that you're doing on the samples as well, isn't there? So maybe we could talk a little bit, maybe in particular the hydrogen, the isotope ratio stuff.
Right.
That it seems like that might be a little bit related to what you've done, what you did previously in Germany, maybe as well.
Not as much, because these isotope ratios are quite specific to. They sit on top of another analysis that I did with GNS.
Sure.
So to explain that quickly, at GNS, I work with my second supervisor, and there I go into the lab and I extract lipid biomarkers, as we call them. So those are remains of leaf waxes, for example, or other plant plant
so
materials.
sort of the waxy surface on the leaves.
Yes.
Those molecules stay.
Yeah.
Over thousands of years.
Yeah. They can be well preserved.
Okay.
And the cool thing is that they incorporate the isotopic imprint and other things, but also the isotopic imprint of the time when they were synthesized.
Yeah.
And with that, so I first extracted and purified the samples and sent them over to Germany. Unfortunately, I wasn't able to go back to work with them myself. And then this lab in Germany were so nice to measure the isotope ratios of my purified biomarkers, and they give that back to me.
Right. So when you say isotope ratios, tell us what those are.
So, so in geosciences, we look at, any shortcut here to think.
Here's where he's desperate for a picture. So we're looking at hydrogen isotopes, right?
Exactly, yeah. So hydrogen, as other elements, has different masses. And depending on the mass, you can get something we call fractionation. So let's say, for example, you have clouds forming.
Yeah.
And we have heavy water and we have lighter water. And so when the cloud forms, preferably the lighter water evaporates better than the heavier. So the heavier stays to a certain amount more likely back in the ocean, for example, the lighter goes up. And the same thing happens then in reverse when we have rain. So the heavier rains first and then the lighter. And so with this, we get over time and rain and transport and evaporation, we get different ratios between those two isotopes, the heavy water and the lighter water. And eventually, we have the signal that rains down on our lakes, or the catchment more specifically, and the plants, they also have their preferred water that they incorporate into their leaves. And then dependent on the plant, on the amount of water available, they form their own isotope ratios, and that's preserved. So we can get information from these ratios about stress, water stress. Like was there not enough water, or was there a lot of water? So we can get a signal just on that already. But then also, where does this water approximately come from? Because we can trace sources and say we have in general more heavy water that is maybe more from the north or than from the south with the westerlies, and with that you can get a sense of what the weather was like. Exactly. Where the clouds were blowing in from, whether they're coming from Australia or from Dunedin or whatever.
Yeah, I mean, we've talked in the past with people on the show about this process, and I remember a long time ago talking to people about it, and that was then, because it's a relatively new technique, a big part of it is understanding. Now, you said that the plant will incorporate this water, one in preference to the other in a certain way, but the way that one plant does it is different to another plant.
Yeah.
And so you have to understand all of that first.
Exactly.
Before you can then apply that understanding to actually what you're sampling. So there's an awful lot of work that's had to go on in the background with all of this. But now that's been done, now you can take advantage of that. And
yeah. That's no.
And you know, and you get the sample and the actual collection of the data. I mean, it has to go to Germany, but for the machine. But actually collecting the information is very straightforward.
Yeah.
Once the machine, as long as the machine's working, from my understanding, is getting the data out. It's very easy.
Yeah.
And very powerful when you know how to do the analysis.
Yeah, I think that one specific difficulty with that technique is to keep an eye on all the possible effects that can influence, or things that can influence these ratios, because, as you said, the plants have their own mind depending on the species.
Yes.
And then the different sources can change. Then temperature changes can also affect the entire thing.
Mm-hmm.
So that really leads to a variability. And that's why it's also very important to have the core dated, because a signal, and that's in general with all signals when looking at sediment cores, they can mean one thing or they can mean 5 different things. It's just the combination of everything coming together that gives a conclusive story. And then the best thing is when you can observe this story in another record somewhere else.
Mm-hmm.
And that's when the magic happens.
Yeah. So again, this isn't something you can do by yourself in your lab.
No.
You have to have access to lots of different people and lots of different toys so that you can get all this different information, put it all together.
Right.
To get to the answer.
And talk to a lot of different people and experts in their own specific fields, which is really cool because it never gets boring. It's sometimes a little bit overwhelming.
Yes.
But there's always something new to play with.
Good. So you're just about finished?
Yeah.
What next? We submit, we enjoy Christmas, we defend the thesis, and then what next for you?
Ideally, I would find a postdoc. I'm very keen staying in science and academia. I'm having, even if there are hard moments, I'm having a lot of fun. And I will return to Germany.
Yep.
And then see if there's something for me there.
Cool. Well, we're just about out of time. Thank you very much, Julien.
Thank you very much
for coming along and talking to us about what you've been up to. Very complicated stuff, but also very interesting and important stuff too, to be fair, in the overall scheme of things. So all the best for what remains of the PhD.
Thank you very much.
Cool. All right. Thank you for that, and thank you everyone for listening. You've been listening to the Science Notes podcast, brought to you by the Division of Sciences at the University of Otago. To listen again, or to find other episodes in this series, go to Otago Access Radio's website, oar.org.nz, or to your favourite podcast platform.
This podcast was produced by OAR FM Dunedin with support from New Zealand On the Air.